All right, so the microbial world and you. What's this going to cover?
Well, the idea is to understand how microorganisms really impact your daily life. That's the idea behind this topic for the very first one we're going to cover in exam one.
All right, so it looks like we have about 6 SLOs that we're going to cover. So we're going to go over ways in which microbes affect our lives, the system of classification, how we name microbes. Then we're going to differentiate major characteristics of each group, right?
Right, so that allows to tell us the differences between them and how to sort them out and how they actually impact us in terms of disease.
Okay, and then there's some key scientists. I know, the microscope was invented by a specific scientist, and they started discovering microbes and whatnot. We're going to go over those, and then contributions made by bacteria. What's that going to be about?
So again, to re-impact, probably 1A in there is that they do more than just cause disease. So what big things have occurred in history that caused changes both in medicine and microbiology?
Right. Okay, and then the last topic is going to be normal microbiota, resistance, biofilms, and infectious disease.
So there we're going to be talking about current hot topics, some of the things that are occurring right now worldwide, and how you should be more or less worried about it or at least aware of how they're impacting human life.
Okay. All right, so this first slide, microorganism versus germ. So germ, that's a word that's thrown out there a lot, right? I mean, it's something we've heard throughout our childhoods, like wash your hands, you got germs on you. But there's a difference, right? There's a certain meaning behind those words, right? Germ is kind of misconstrued.
Right, and the issue here is that the germ literally means any type of cell that grows really, really fast. So that includes viruses, that includes bacteria, although viruses are not quite cells, but anything that usually divides really quickly can be referred to as a germ. We actually use the term germination in plants as a way when plants are actually starting to grow. So it's not a really good term to use these days. And so we kind of stray away from that and we start sticking to the new term, either microorganism or microbe. Both of those are pretty easy to use.
Okay, so I'm curious about something. Viruses, do those fall under, those are microorganisms, right?
Correct, but they are not cells. So keep that in mind. And
Okay.
we'll have an entire lecture devoted just to differentiating those two.
Got it. Okay, so using microorganisms is a little bit more appropriate
Right.
for us,
Correct. In our field, that's the terms we should use.
not germs. All right. So I know there's just countless microbes out there, but not very many of them are actually disease causers, right?
Right. So one of the common misconceptions also about why it's important to study this, is because only about 3 to 5% of them actually cause disease. The rest of them either don't really care about us very much, or some of them are actually trying to help us out.
So that's probably news for a lot of people that they don't actually cause as much disease as you think.
Right.
So some of the things that microbes do, I know on this slide we've got they decompose organic waste, they support the ecosystem by photosynthesis, so produce oxygen and whatnot, produce industrial chemicals. See, I didn't know this. This is pretty interesting. Ethanol, things like that you wouldn't expect, those are all produced by microorganisms, right? Those are like byproducts.
Right. So one of the things again to remind you of that 95% that are not out there to harm us or don't cause disease is that some of them have a lot of medical or industrial applications. So you just mentioned a couple of them that are listed in there. The most basic ones, you mentioned the production of oxygen. Believe it or not, the transformation of oxygen into the planet was not caused by trees. It was actually caused by bacteria. Another common concept is that they work as biorecyclers, so they help clean up the environment. So that means anything from destroying anything from our paper waste, plastic waste, and even extended to cleaning oil spills. And probably one of my most favorite topics to talk about is the production of food. Industrially speaking, anything from fermentation of things like alcohols and acetones and bleaches and food items like bread and cheese are all standard things that microbes contribute for our life.
Interesting. All right, so E. coli, we were talking about this earlier. That's a bacteria that has a lot of like negative connotations associated with it.
Right. And the issue that is, again, common misconception. Yeah, it's found inside your intestines, usually helping you kind of with your digestion a little bit. But if it leaves that area, it can cause some major disease issues, especially at food industries. But here, you'll notice that no food present. In fact, what you're seeing there is a giant flask with the color blue. And the reason behind that actually falls into our next slide, in which probably one of the most surprising effects is for people to realize that their clothes, most of the items that they're wearing right now, probably produced by microbes also. What are we talking about here? Things like denim, so your classic Levi's that everybody's talking about. The bleaching of the cotton comes out a little kind of different colors, not quite bright white that most people assume from cotton, is done by bacteria. The colors that are produced for your items of clothing also can be produced specifically by E. coli, but even more, kind of the effects that you get in your clothing from the bleaching effect, the stone washing effect that you're talking about, also done by microbes. And one of my favorite ones is to remind you that even the buttons that you're wearing right now are probably produced or at least synthesized in part by bacteria as well. So they do contribute a lot of things aside just trying to kill you or cause you some sort of disease. So knowing about these things is one of the important facts of this subject.
All right. So next we go into knowledge of microbiology. This one was kind of interesting to me. I know this has a lot to do with temperatures, and throughout the class we're going to be culturing, bacterial cultures, and there's specific temperatures that we keep them at. And that's also taught us how to preserve food, how to prevent bacteria from growing on things. We keep our foods in the fridge, keep them cold and whatnot, and that slows the growth of bacteria, right?
Right. So again, one of the important factors of this class is, yes, understanding how they can harm us, but also understanding that if we know their properties, we know how to control them. And so the idea of knowing that they grow roughly at around 37 degrees Celsius most of the time, body temperature, that means that if we change that body temperature, like fevers, or if we cool things down like in a fridge or in a freezer, they will stop growing, which means you can actually make your food last longer. Or in the same way as like a fever does, it reduces the effect of an infection. And so what we're going to do in the lab, like you just mentioned, is we're going to learn how to control that, specifically by being more or less clean. A term that we're going to use for us is a septic technique, in which we'll learn how to transfer these organisms by reducing the chances of contamination of yourself, others, as well as your environment. So that's going to be a really big deal. So since we're all going to be healthcare providers, this is an important fact of not contaminating our patients, each other. And so we're really preventing the dissemination of disease.
Right. All right, so classification and taxonomy is the next subject. And I remember this one was a little bit confusing for me back in the day, because there's so many names that that are out there. There's so many bacteria that are out there, and we've attempted to name pretty much all of them.
All the ones we know so far. Now, here's the deal behind this one. The reason why we actually do this is because, like you said, there's a lot. And so how do we keep track of these, and how do we know which ones we're dealing with? And more than anything else, how do we treat them in the case of disease? That's the biggest deal. So the field of classification sorts out our organisms, usually by genetic relationships. And what we know now is, for example, that we have sorted out that these three large categories called the three domains of life, which are bacteria, which we're going to be talking about a lot, archaea, which are their cousins that have some differences between them, and then the eukarya, which is pretty much the one that most of us are familiar if you've ever taken a general bio class. This is the ones that include us, animals, plants, mushrooms, and all that good stuff. So that's more or less where we're going to start. But for now, right now, our important piece is to kind of introduce you to the subject on how to name things.
Okay. So each organism has two names, right? The genus and the specific epithet.
Right. And if you're wondering, that's what most people call species. So some people abbreviate that term. So if you ever heard the term species, the correct term we use here is specific epithet. But there's some rules on how to use that.
So that's why we see like Escherichia coli, there's the two names there. So the genus is Escherichia, and then the specific epithet is coli.
Right. So the species would be coli. Right. Now, here's the interesting thing about that is that because of the fact that most people abbreviate it as E. coli, there's tons of other organisms that start with E and end with coli. So it doesn't quite help us that way. So it's kind of important to kind of spell it out the very first time you use it. And here, there's a couple of rules that you should always remember. This we use in the lab, in the exams, and we will kind of grade you based on this. So it's important that it's always italicized. And so unless you're really good at doing that with your hand taking notes, it's easier if you underline it. So that's usually the default. So always underline both terms, the genus and the species terms, and making sure that the very first time you use it, that it's fully spelled out. Afterward, you can actually kind of use that and abbreviate it like we're normally familiar with it. However, one of the important things to know is that because microbiology and all these names have been around for a while, is that some of these names, like you said, Escherichia specifically, comes after somebody's name. So it's actually done to honor a scientist, Theodore Escherick, who's the person that was working with it. But that doesn't really tell us anything about that organism at all. And so not a very good mechanism of naming things.
Yeah.
On the other hand, the term coli usually relates to the term colon, so we know roughly where to find it.
Right.
So nowadays, we kind of stay away from naming things for the sake of just giving fancy names. Instead, I can give you the other example of Staphylococcus aureus, which here, it's very descriptive of what it does. The term staph here, meaning that it's clustered, actually means grape-like in Greek. The term coccus here, meaning that it has a little spherical shape or round bubble-like shape. And the term aureus comes from the term aurum, from gold, which means it's kind of gold colored. So this one gives you a much better idea of what that organism is. So reminding you that, again, we're going to use the terminology, make sure that it's a two-term system. This is called the genus and the species, or specific epithet, that it needs to be underlined or italicized if you're really good at that, and that the very first time you ever use it, it's fully spelled out, and then after that, you can actually use it in its abbreviated form. So the first time you say Escherichia coli, that's correct. After that, you can just abbreviate it as E. coli.
Got it. All right, so this is going to go over what's covered in microclass. So you were talking about archaea, bacteria. Those are two of the main domains. Archaea, those are like the ancient, that's what I associate with it from what I remember. Those are older bacteria, right?
Well, kind of bacteria. They're not actually bacteria, hence why they're separated into other terms, but most people see them as such, so it's okay to kind of talk about them in that way. They look very similar. They have similar properties, but as we're going to mention in a moment, there are some key differences. Okay. But that's not the only things we're going to see. Believe it or not, in microbiology, there's plenty of other cool stuff to actually see. We're going to talk about mushrooms, so in this case, the group of fungi. We're going to talk about these unicellular organisms that are kind of mini animals, things like that, called the protists. And then we're definitely going to look at viruses, and then we're definitely going to look at things like prions and virinos and other terms that probably you haven't heard of quite yet. And probably one of the most fun ones, and usually gets to be a little bit gross, are the animal parasites. So one of the reasons why we highlight it here is to ensure that we get just a very small taste of what's going to happen later on. And we will literally spend many lectures talking about all of these in full detail. So let's go with the first one.
All right.
So you asked about archaea, right? And you said the term ancient, which was accurate here, just like the term archaeology. The idea here is these are organisms that are unicellular, that don't have a nucleus. The term prokaryotic comes into play if you've heard that term before. But the most unique thing about them is that they do not have cell walls. They don't have that extra layer of protection that bacteria normally do have.
Okay.
Another unique thing you can remember about archaea is they all divide by kind of popping off from one another in kind of halves, a term called binary fission. But the cool thing about them being ancient is that they lived or survived until now as well, is in these so-called extreme environments. Places that are normally too extreme for us to live. It's not extreme to them, it's extreme to us, right? And so we're talking about anything from, thousands and thousands of feet below the ocean, and volcanoes and sulfur vents, places that typically humans and other animals don't hang out more often. These guys, this is their specific highlight, right?
Yeah.
And so we'll talk about what we call, for example, the extreme halophiles, ones that like really salty environments, ones that like really hot environments. These are called thermophiles. So we'll spend an entire lecture covering those guys.
Okay. All right, so on to bacteria, prokaryotes. So is that like a specific type of bacteria, prokaryotes, or are all bacteria prokaryotes?
You got it right the second time. In this case, we're talking about that they all are bacteria prokaryotes. The term prokaryon here means that they do not have a nucleus, that their type of cell, their genetic material, typically DNA, is kind of free floating around in a little cluster, as opposed to the other organism we'll talk about, the eukaryotes, that do have a container for it. So that's the biggest difference that you have in there. The other one that we just mentioned in Archaea was the fact that they do have a cell wall, so they do have an extra layer of protection, but they divide just the same as they do in Archaea. But these ones happen to live in our environment as opposed to extreme environments. And so we'll start differentiating those a little bit later.
Okay.
And so these are your classic ones. These are like your E. coli that you talked about earlier, the Staph aureus that we mentioned a little bit earlier. Those are those basic guys.
All right.
So as we go, we'll start giving you some more examples and pretty images of what those are as we go on.
All right, moving on to fungi. So fungi are eukaryotes. So they do have a nucleus, right?
Right, so that means their genetic material is contained within a nucleus. Now these guys also have walls, so very similar to bacteria in that case, except that they do have a nucleus as well. Here, the way that they reproduce is a little bit different. They don't kind of split into a couple of cells. Here they undergo very similar mechanisms that are kind of asexual or sexual types of differentiation.
Mm-hmm.
We'll talk about those later. One of the unique things about fungi and the groups of mushrooms that are out there is that they're using food that comes from some sort of organic source, meaning some sort of living thing, or typically more than anything else is as they're dying. And so that'll include all our typical molds, your classic mushrooms that everybody's familiar with, and we'll get to see how they grow, why they grow, where they grow a little bit later. But not to forget, one in particular is that part of that group are the yeasts, the same yeast we're talking about for brewing beer to baking bread. Those also fall in that category. And in case you were wondering if you've ever seen one, your probably stereotypical one is that classic bread mold. So I'm assuming you've been at home, let some bread get a little bad, and see that wonderful set of colors popping up. That's your classical fungus right there growing on that guy.
All right, so Protozoa. Those ones are kind of interesting. That's more moving into like almost little animals, right? Like they've
Exactly
got more
it.
parts and
Right, so these get a little more
complexities.
complex.
Yeah.
Right, so one of the terms you'll learn a little bit later is that the term protozoa means early animal.
Mm-hmm.
And even though they're not animals, so let's not confuse it there, but they are eukaryotic, which means that they're typically multicellular, like animals, or in this case, they move like them more than anything else. And one of the unique things about protozoans, part of this group of protists, is that really they are unicellular, but they move a lot.
Mm-hmm.
And so we'll talk about how we classify these guys and sort them typically on how they move, how they kind of swim around and turn and twist and squish each other in order to look for food or look for other ways of looking for energy sources.
So I'm curious, would a white blood cell fall under that?
Unfortunately, no, because our white blood cell here is a human cell. And humans, even though we are eukaryotes also, they're definitely a multicellular organism. They're part of humans, right? Protozoans, on the other hand, easier example I can give you is your classic amoeba. Everybody's heard about it typically, especially if they took some sort of gen bio class in high school or in junior high, and they saw little things swimming around in pond water or scum and stuff like that. Those are the guys we're talking about in terms of protozoans. These ones don't get complex. They kind of stay kind of small, kind of unique unicellular ones. They are still eukaryotic, but they like to move. They like to swim. So we'll get to kind of look at those, especially in our class, because of the ones that cause disease.
All right, so moving on to algae. Algae, I mean, they're kind of like plants, right? Eukaryotes, but, I mean, and they have cell walls, and they use photosynthesis, so they
Right.
produce oxygen, right?
Yeah, so I was talking about a little bit earlier is one of those common misconceptions about the whole thing of how do we get our oxygen in our air. This idea that trees do that, yes, definitely trees contribute it, but the biggest contribution to our planet was done by algae. This is one of the important reasons why we look at it, is that algae are also unicellular. These are also eukaryotes, but these are kind of plant-like, as you mentioned earlier.
Mm-hmm.
They're not quite plants, but they're similar to them in which, again, they carry out this so-called photosynthesis and another process called photophosphorylation, in which they use sunlight or any light source to produce their own energy, their own food, and as a byproduct, kind of as a waste product, they will produce things like oxygen, which is what we need in our planet to survive. So normally what it means is that when you look at algae, they're kind of going to be green-like, very much like most plants are. That doesn't mean they're quite classified as plants. So we'll take a look at those a little bit later too.
All right, so now moving on to viruses. Those are pretty interesting. They're a lot smaller than bacteria generally, right?
Significantly smaller, usually by 100 to 1,000 fold at least, so much, much smaller. And the key feature here, as you notice in the first bullet there, is that they're acellular, meaning these do not involve cells. So there are no walls, there's no membranes that we normally talk about when we're talking about bacteria, archaea, and eukaryotes. These guys are much more simple. They usually have a little container made out of protein, and their genetics are usually contained inside that little tiny little package inside of them. Sometimes they have a little bit of lipids around them, but they're very, very simplistic. And so one of the key features that we'll talk about a lot later on is that because they're not cells, because they're acellular in this case, and they do not have the same properties as your bacteria and archaea, is that things like antibiotics don't work on them. They have no effect on them. They don't really have the same type of use in terms of medicine that it would apply to your classic E. coli or something like that. So when we talk about viruses, we'll talk about multiple examples. Most people think about things like HIV, HPV, HSV, and all these things that end with V, indicating a virus, right? Yeah. So we'll spend a good amount of time in an entire lecture just kind of highlighting how they actually cause disease. And so we'll have some fun with those a little bit later.
All right, so multicellular animal parasites. So these are kind of gross, right? But we get into talking about a lot of those. So those are eukaryotes. They have cells that contain DNA inside of a nucleus.
Right.
And then there's all kinds of parasites, from fleas to worms and all that.
Right.
Fun
so
stuff,
again,
right?
another good chunk of time. This is the parts that get a little bit gross to people to see, because these are the ones you get to see. These are, you can see them with your unaided eye. No microscopes are needed. And most of the time, things like worms are the easiest way to observe, and things like you said, ticks and fleas and all of those fall into that category because at some point in time in their life, at some sort of stage in their development, they were actually microscopic. And so that means that they get included in our subject. And so we will spend a good chunk of time discussing specifically a great amount of time worms. And so their variations from flatworms to roundworms, and some of the diseases that they carry inherently, or some of the things they carry inside of them that they can also pass on to you. So we'll have some fun with those, especially when we learn and classification. So that's kind of the overview of a lot of the things we're going to see much later on in the class once we get past the introduction subjects.
All right, so brief history of microbiology. This is kind of interesting. This covers a lot of the scientists that contributed to microbiology, right?
Right. So again, here I'm going to emphasize very, very importantly is that it's kind of like history. It's important to know it, but this is not a history class. So what we're going to do here is more or less mention some of the things that allowed us to advance in micro, but we don't really need to know the names. We don't really need to know the dates, so we won't get tested on that. I'll mention them as we go as examples, but the idea is here is what happened in history that allowed us to understand and really advance the field of medicine. So the contributions are what's important, not necessarily their names or their dates. That's something that anybody can just do a quick online search and find more about it.
All right, so origins of micro. I know it wasn't all that long ago that people didn't realize that there's these microscopic little organisms everywhere, right?
Right, and so it all has its origin, and the biggest issue here is a microscope. So we usually go about 400 years back or so roughly around the 1600s, in which a few different scientists started kind of getting a little curious and realized that, yeah, there was life smaller than what we can observe. So what we're going to talk about here is just, again, reminding people about the names, but we don't really need to know them. We need to know their contributions. Biggest thing to remember here, definitely on the exam, is this subject called cell theory. And thanks to several different contributions from scientists, there's key points that everybody needs to know about cell theory. What it means is that in terms of our subject, is that all living things are made up of cells. That's tenet number one, so that's important bullet number one of cell theory. that cells are what we call the most basic unit of life. So the smallest living thing that we consider alive is called a cell. And as a consequence of how biology works, is that cells have to come from other cells. They don't poof into existence. They more or less need a predecessor cell or a parent cell to give rise to more cells. So putting those three things together is what we call cell theory. Now, what we're going to kind of talk about here is more or less some of the guys that did that and what were they doing and how did they get to this concept of cell theory. And so for that, we're going to talk about the microscope itself. So the first piece here is two scientists that we have. Leeuwenhoek, which is the gentleman that more or less started working with little glass beads. And by putting pieces of copper together, he was able to kind of scratch off stuff with little piece of nail. And by raising it up to the sun, he would see that things were moving. And so little glass beads, that's all it took. And so what he sees is what he eventually calls animalcules, or tiny little animals. And that was kind of the beginning of microscopy as well as microbiology right there. But he wasn't alone. Another gentleman by the name of Robert Hooke, roughly around the same time, kind of instead of using beads, he started using lenses. He started actually fashioning glass lenses, and the same idea, magnifying things. And this is the very first time that we get to publish things in a book called Micrographia, in which he would draw things. So what you're seeing here are not actually pictures. Those are drawings. Wow. No cameras, so in case you're wondering. And so Robert Hooke painstakingly drew out in detail what he was observing. And this is the very first time we get to use the term cell. The idea of the history behind the cell is because what he observed, as you're seeing in there under a piece of cork, which is what you see drawn there, is that it looked to him like it looked like a convent, like little rooms, squares. And so he referred to them as cells, and that's really where that origin of that word comes from, right?
Okay.
What is here is important is that these two gentlemen specifically really revolutionized the field of microbiology. Before them, we really didn't know what was kind of out there. Thanks to lenses, thanks to glass beads, and this idea of magnifying things, we were able to see that life did exist beyond the scale of our eyes. So the invention of the microscope is the biggest deal. So here we have to talk about some terminology. It's important to know the differences. First of them is resolution. And what resolution really means is that microscopes resolve things, and that means that they're able to make you tell the difference between two objects. In other words, as you start getting smaller and smaller, how do we know that one object is one or two? And so microscopes, with their resolution, they're able to separate things out into two or more objects technically. The way they do that is by using a property of light called refraction. So don't confuse the two of them, two separate letters there, in terms, sorry. Is refraction means bending light. So you refract light, and what happens is the light that we can see bends and it refracts off the objects that we're observing, and we are able to magnify them or make them bigger to observe. Now, the only issue with this is that refraction is limited by the availability of light, meaning that we can only resolve things or see things separately as long as we can see the light. So if we can't see the light, things like UV or infrared, we can't see the object either. So it's kind of an important thing to understand is that our vision using a microscope is actually limited by how much light we have available to us at that time. And for that, we need a quick refresher of physics. The idea of what everybody learns about the rainbow spectrum and how many cool colors are in there, that's as much as we can see. So this very small range of light that exists, that's what our microscopes can show us as well. Anything above that wavelength, anything below that wavelength, can't really see. We're kind of blind to it. So we have a very narrow range of how much we can observe with that. So one of the issues that we have is that our microscopes, as we start with the most basic ones, what we call light microscopes, usually only come with one lens. And that means that they can usually magnify things up to about 40 times bigger. Now, the problem with those light microscopes is that most of the things that are smaller than our eye can tell are also transparent, which means that even if you magnify them 10, 20, 30, 40 times, still can't see them. So normally that means we have to add a little bit of color for background. So this is called staining, a topic that we'll deal in lab a lot later. So we'll learn about specific stains like simple stains and gram stains later to be able to observe them a little bit better. But somebody got smart and decided to put two lenses together, and this is the idea of a compound microscope. So in which you take two lenses and kind of put them in series, and now each lens is technically multiplicative, meaning that each power of each lens can multiply the other. So this is what we call a total magnification. So the power of one times the power of the other one gives us how much more we can see by compounding those two lenses. Cool thing about that, yes, we can see a lot smaller, roughly about 200 nanometers or so. But even then, that's kind of the limit of the compound microscope. There really isn't much beyond that. So these are the ones that we're going to be using in class. So very similar to those guys. And again, we'll learn about their anatomy and more details on how they work. We have some really cool ones we get to play with. Sometimes they come with one ocular, two oculars, that kind of thing. But they all do the same job. They'll magnify things up to about 1,000 times, and we'll be able to see roughly about 200 nanometers. But how do we know that there's anything smaller than that? For that, a little bit more physics. That means that there's a brand new set of scopes that are called electron microscopes. Now, for those of you who remember your general chemistry, we can't see electrons, obviously, too tiny.
Right.
But we can use technology to use that. So things like computer screens and film and stuff like that. And so here, very much like light, we use electron beams instead of light beams, and we use those to magnify the object. So we can use a computer screen or something like photographic film to do so. Now these type of electron microscopes come in two large flavors per se. These are called the scanning and transmission microscopes, and each one of them has some significant amount of power. Your compound scope magnified 200 times, 200 nanometers. The scanning one can magnify up to 10 nanometers, which is about 20 times better. So whatever you're seeing in your regular scope that we have in the lab, 20 times better than that. The transmission one, or transmission electron microscope, also abbreviated TEM, magnifies things to about 5 angstroms, roughly the size of about 5 individual hydrogen atoms. So we can get really, really well into the area of chemistry and their properties. Now the problem with electron microscopes is that they're incredibly large, and so they usually take up several tables, if not rooms. But once they get them to work, we can actually see things like pollen grains to viruses to exquisite detail of the intestinal flora and our microbiome and what's growing in and inside of us, to individual cells just in perfect images. Now the even better thing about this is that the highest level of these microscopes, what we call usually atomic force ones and other fancy names, is that nowadays we can actually see individual atoms moving together. So we can actually see compounds and the bonds that are occurring between them. So we have reached the level of technology in which we can actually see chemistry happening. So just in case you want to see a little bit of the scale, we have the ranges kind of posted on the slides. That way you can see what a light scope can do versus a compound scope or electron scope, and some of the specialized ones that allow us to see things like atoms and bonds. So not to kind of dissuade you, but we're kind of on the right side of that spectrum, in which we can only barely see things like little bugs and fleas and sometimes really, really, really large protists. But everything beyond that, we're going to need the help of a good scope. So that's why we have the lab. That's how we get to discover all the cool things that microbiology actually does.
All right, so back to microbiology origins. So spontaneous generation, this was like a concept that, this is like an early concept that they had back in the day, right?
Right, so this fun one of the idea that things spontaneously came out of nothing. So hence the term generation, right? Mm-hmm. Now, the problem with this is that now we know that to be inaccurate, but for a very long time, lots of people actually sacrificed their life for that. They actually died for proposing that life came from something as opposed to from nothing. So spontaneous generation more or less meant that life came from non-living or non-existing things, and that was the term that these group of people used to follow. And then the group of scientists that revolutionized that were the ones that believe in something called biogenesis at that time, the idea that life had to come from other life. And this is, again, the same thing we talk about in cell theory. Cells have to come from pre-existing cells. Now, How do we know this exists? This is that history that I was mentioning earlier. We don't need to necessarily know the names nor the dates, but it's kind of important to know what these people did in the past to let us know that really it's not spontaneous. There are reasons why they exist and how they come to be.
So didn't they used to think that there were actual like ingredients to create a living thing, right? Like,
Right, so they came up with various different origins, from magic to wind blowing in the right direction, things like that.
things
all
in a
over-
bucket. Wouldn't they put things in a bucket and say a rat would show up, but it was actually food and the rat would show up
Right,
to eat the food?
so
Something like that.
people have
It's kind
changed
of-
their ways of
kind of
thinking. Thankfully,
interesting, yeah.
we've advanced since those times, so that's a good idea.
Mhm.
Probably one of the most critical ones here against this idea was an experiment that was proposed in the mid 1600s by a man named Francesco Redi, in which what he did, very similar to your rat concept, is that they would put little pieces of meat inside some jars, and the idea is that they would see that soon things like maggots and worms were growing on them,
Right.
and that was immediately saying, hey, look, spontaneously popped out of nowhere.
Mm-hmm.
Now, at that point in time, people didn't realize that maggots were the result or the basic babies of flies.
Right.
So they didn't know that at that time. So more or less they said, look, magic was producing these little baby worms. In reality, what it was is that a fly landed on the meat, laid its eggs, and eventually gave to those. But at that time, nobody knew that. So a very, very simple experiment was to put two jars with that same meat, but covering one of them with a thin net. So it was more or less open to the air, but bugs, like flies in this case, could not reach it. So what happens, you have some covered jars, some uncovered, and what you'd find out is that the maggots would actually appear directly on the meat of those that were uncovered, but the ones that were covered, perfectly clean meat, but you definitely see the maggots on the cloth. So clearly it wasn't the actual meat that was causing the maggots, it had to come from somewhere else.
Okay.
So a very kind of unique beginning of this idea of that no, maggots didn't come from nowhere, they weren't spontaneously generated. So these were the beginnings of arguments against spontaneous generation. Problem with this, they didn't really like this person and they actually sent him to jail. So, now, along with this, to kind of argue with what Francesco Redi had done, a gentleman by the name of John Needham took the same idea, but instead of using meat, they used broth, so a good old stock, right? And same concept, they would actually boil it as a way to indicate, look, fire should kill things, as it mostly does. And so they would heat up the actual flasks with the stock inside of it, and then they would say, look, things still grew. And if that was the case, it's spontaneously generated because the heat should have killed everything that was on it.
Right.
Problem is like everything else, if you don't really cover it, things can still land on it. Or alternatively, if you heat it and don't have it covered beforehand, there's still a small amount of time for things to land, and yes, things will still grow. So you can see that there was a little bit of logic that needed to occur there, but people nonetheless will believe whatever they want to believe. So this is why people continue to challenge them. Now, roughly around the same time, a man by the name of Lazzaro Spallanzani, same idea, took the same broths that Needham was doing as a way to argue with this, but one key feature. Here, the flask was kept sealed and then boiled. So that meant nothing could get in there first. So if he cooked it, no growth. And again, going towards this idea of no spontaneous generation. So little by little, these are the experiments that occurred. Now, we're telling you this story because eventually, probably one of the more infamous of them all, probably a name that everybody hears about, is Louis Pasteur. Now, notice that the dates, we've gone from the 1600s to roughly the 1800s here. Lots of experiments. What did Pasteur do in this case is same idea, but with a small twist. So it's easier to show it more into the diagram. And what Pasteur did is by using the same concept of flasks, broth, meat, that kind of idea. The only difference is he twisted the entry point. The idea would be simple, that we can keep it out in the open so that life can occur. But if you bend the flask, that means that things like flies or dust particles or anything that can contaminate it can't quite travel through that neck. So boil it, sterilize it more or less, and he found out that nothing would grow. So it was out in the open, but no bacteria, no bad stuff, no flies could actually reach it because of the long neck. And now he was able to show that definitively it wasn't the fact that it wasn't closed or that it was open. It was the fact that the fire actually sterilized everything in there and nothing else was landing. So a very elegant way of showing that.
And if I remember correctly, some of these flasks are still around today, right? And they still haven't grown.
Right. So a little bit of history
microbes.
kind of shows as a way to kind of promoting this idea that nothing can grow with them. Very simple concepts. You can do this at home if you really have the access to the glassware. So kind of fun to do. All right. So going around again with this concept of those that came before us and what's the big deal. These people made important contributions. Now, what is the impact behind them? Clearly, Pasteur kind of opened up the road of more scientists to discuss this idea of small organisms taking over and causing disease and so on and so forth. So we're going to mention a few of them. Amongst them are people like Joe Lister, Robert Koch, Edward Jenner, and those are, again, not necessarily the important idea of knowing their names or even the years, but what their work has done.
Right. And there's like, modern things associated with those names that you probably recognize, like pasteurization. Isn't Listerine associated with Lister?
That's exactly it. So kind of let's get into those very quickly. So obviously Pasteur was able to kind of demonstrate that heat would actually change things, and believe it or not, he's responsible for producing this concept of fermentation, or the idea of fermentation, and how to transform things like wine and beer. So he gets a lot of that credit behind that. But this idea also of preserving food came from by using flames. So you can actually preserve food by just heating it up, as long as you didn't give it access to that. But you're right, he kind of led the team and other people came about. This is the concept of the germ theory of disease, in which more scientists started to come about saying, look, we've studied what Pasteur and his predecessors have done, and we realize that magic is not killing people, it's more microorganisms. So you mentioned Lister and Listerine. Well, the gentleman associated with that was Joseph Lister, that started using a compound called phenol. We'll talk about that later. A fairly dangerous compound now, but back then was used as an antiseptic. So used in surgeries as a way to clean wounds and prevent infections, worked really, really well. So unfortunately, his name didn't go into exquisite fame, aside from the fact that we now use it in mouthwash and Listerine. So that's where that name kind of applies. That other people introduced in the mid 1850s roughly is this idea of washing your hands, something that we all now take for granted. Every parent tells you to do so, but that wasn't something that was done a couple of 100 years ago, and that led to lots of people dying. So it was key events that, again, we take for granted now that are very, very important to this date in terms of medicine and health. Along those lines, Robert Koch, which creates something that we now know as Koch's postulates, a term that we use in all clinics, was this idea of proving, more or less, as best as you could back then, that one particular organism causes a disease. Now, he actually worked with Bacillus anthracis, the organism that causes anthrax. And so what it was here is that Koch actually demonstrated as a way to test how many organisms would cause specific diseases. What does that mean? Is that his idea was to prove that one organism would lead to a specific disease, and he usually did it in rats. What he would do is find something that was ill, extract it from it, grow it, pass it on to a new one to see if it got the exact same disease. When that actually happened, he would extract it again to confirm that the first one matched the last one. And so this is known as Koch's postulates, and we'll actually be doing this in the lab a lot more. Okay, so we'll start with those, just give it a little bit of time so we can expand upon this guy.
All right. So moving on to vaccinations. So this is kind of an interesting subject. Vaccinations, we actually inject a weakened version of viruses and bacteria.
That's correct.
Into our cells.
Now, it doesn't need to be injections anymore. With the advances of sciences, now we can actually do it orally. We can get little drops for you to take, so we've actually changed the delivery. But most of the time, they're pretty much injections. Now, the reason why this guy's important, again, part of the history, is because what happened. So again, in the late 1700s, early 1800s is that somebody decided that we discovered by accident that by giving inactivated or weakened versions, like you said, of certain organisms, we could get a little bit of protection from them, a subject that we will learn at the way end of the class called immunity. And so this is another big deal, again, that has happened over time, the ability to save lives, and we've contributed significantly the reduction of disease simply by this concept of vaccination. So we'll get to explain that a lot more later on in the subject. So that's one piece of history. Another big history associated with that same idea of vaccination are drugs. And this is a big deal simply because obviously this is a big industry, a big impact in terms of society and economics. But the idea is that we were able to obtain drugs as a way to treat medicine and disease. What does that mean is that even as far as 5,000 to even close to 12,000 years in our history, people were using things like leaves and chewing on pieces of trees, and they figure out that they got certain benefits. This is how we discovered aspirin, for example. But all that history led a gentleman by the name of Paul Ehrlich to think that maybe, just maybe, there is a drug or a compound or a piece of medicine per se that could target a specific agent that causes disease, a pathogen. And so the term magic bullet was coined here, the idea that if we take a bullet, a drug, a medicine, that would target the bad guy, a bug, a bacterium, a virus, and kill that thing without harming the host. Now, this popped up in the early 1900s, and so we only have roughly about 100 years worth of history in which we've managed to develop the very first antibiotics, which we'll talk about also in the class, and then a group of drugs called antimicrobials, because antibiotics only refer to things that work on bacteria and also are produced naturally. Antimicrobials include things that are produced synthetically. So there's a large bit of history there also happening. But the very first big deal happened with the discovery of penicillin. And this was a drug that is produced by a fungus, by a mushroom called Penicillium. And it was all discovered by accident. So again, a little bit of history, not that this was targeted, it just serendipitously occurred in the 1930s, in which a gentleman returned to his lab and realized that his experiments were contaminated, something we want to prevent in our class, right? And he realized that nothing was growing around this fungus. And he realized that there must be something inside that fungus that's preventing the growth of something else. And so from there became the history of isolating the substance that now we know as penicillin, and the birth of antibiotics and modern drugs and therapeutics. So again, a lot of history associated behind this.
All right, so next topic, the benefits of studying micro. So everything we learned in this class actually kind of changed my conception of how things work, how diseases are transmitted, how we can protect ourselves from certain diseases. Is that mainly what this topic's going to cover?
Well, that's what the class is going to cover a lot. But again, not to forget that only about 5% of them can harm you. A good 95% of them are really not doing much or kind of helping you. So there are certain good things to know from it. One of them, again, we mentioned earlier, is the fact that we can use these organisms to help us, a subject known as bioremediation. So if you ever wonder what happens after your sewer gets emptied in your septic tank, or what's going on after your toilet flushes, or your sink is done with all that stuff, or every trash bag that you throw away, we actually employ organisms to more or less recycle these, a process that we know as bioremediation. Now we've taken that to the extreme, to the point that we now synthesize certain organisms. We've modified them so they can break down things like oil. So anytime barges crash in the ocean and lots of oil spills are spilling millions and millions of gallons of oil from these tankers, these organisms are spread on top of it, and they'll break it down into simpler substances that are less toxic. So this subject of bioremediation in a field of microbial ecology, in which organisms are used to help the environment, is a big deal. But that's just one side of the spectrum, good benefits. A different side of this story is that we can employ this in farming. So California happens to be a large agricultural complex, and so this day and age, we're worried about safer alternatives on how to treat our plants and our food, our carrots and our potatoes and things like that. And here we have organisms that naturally produce substances as insecticides. So again, the term here being naturally produced, there's no synthetic drugs, there's nothing, that is being man-made or human-made per se. Here, the organism produces it, and it actually harms things like pests. So typically things like caterpillars are destroying leaves and trees and things like that. An organism known as Bacillus thuringiensis produces a toxin that is harmful to these pests but doesn't harm our animals, ourselves, and any other plant. So these are other versions of using microorganisms towards our benefit. But even better, the industrial application. So I'm pretty sure there's plenty of food out there that you're familiar with that bacteria produce them.
Right. So here we have some images of, looks like cheese mainly, right? So
I'm not going to deny my bias towards cheese at this point in time, but keep in mind that that's just one piece of food. Think about how many fermented products from milk that you're familiar with.
Mm-hmm.
What other things you know that are derived from milk that you consume every day?
Yogurt's probably a big one.
Definitely.
And if we look at yogurt under a microscope, are there? I've heard there's like active bacteria still in there, right?
It depends on the type of yogurt. It depends on how it's produced. Yes, bacteria mainly are producing these. Mm-hmm. However, depending on how it's packaged or pasteurized, that term you used earlier, right, some of them might be dead already. And so when you hear terms like probiotics and Greek yogurts and things like that, they have that. But don't limit it to those. Things like sour cream, things like cottage cheese also vary behind these things. So it's not just limited to that. So there's a big food application and industry there. But we won't stop there. There's a much bigger aspect behind biotechnology and microbiology coming together, in which we now take advantage of the properties of these microorganisms for the betterment of humankind. So anything from producing new antimicrobials to helping in treating disease and subjects like gene therapy by using viruses. So there's a much bigger power associated with understanding microbes and microorganisms. But probably the one that you mentioned beforehand is the one that we'll kind of expand a lot later, especially during the immune system, is the microbiome. Mm-hmm. So remember what these guys do.
Yeah, so this is something kind of bizarre. I know that the microbiome makes up more of you than you.
That's absolutely right.
So there's more cells that aren't your own on you and in you than there are that are your own. Like if you were to remove all of your microbiome, it would cut your weight in half, right? Or more.
The numbers are in half, yes, or a little bit more, but not the weight. The weight is about 10-15% of you.
Interesting.
But in terms of numbers, you have about 40 to 80 trillion cells that are human cells per se, that are you. Mm-hmm. But there's also about 40 to 100 to 150 trillion cells that are bacteria.
Right.
So they definitely do outnumber you. So, but the idea behind them again is that they're on you, inside of you, around you, without causing you any harm. Mm-hmm. And most of them are just there doing their own thing, and some of them come with some protective effects. Some of them protect you from other infections. Some of them help you process your food inside your intestines. So this idea of the microbiome, also referred to as your microbiota in case you're wondering, help you protect from other infections, so things landing on you, so they're there first, so they prevent things from growing there further. Things like E. coli contribute inside your colon. They help you produce certain vitamins as you are consuming and extracting nutrients. And more than anything else is that some of them produce certain substances on your skin and most mucosal surfaces that prevent the growth of other organisms. So they do have a positive effect on you, but not necessarily all good. Definitely some of them can be bad, and probably the most infamous of them all is the reason why you brush your teeth. I assume everybody should be brushing their teeth these days, right? And this concept known as a biofilm, in which certain microorganisms will kind of work together and form these specialized groups of organisms called biofilms. And plaque happens to be one of them, but we get lots of these examples growing on things like metal and plastic, all over medical equipment. So one of the reasons why we have to constantly sterilize and clean these, because they attach themselves really, really strongly to these surfaces and can cause some severe diseases. So there's two sides of that story. Now the last piece we're going to cover here is a concept known as EID, or an emerging infectious disease. What we're going to mention here is a very, very brief story of things that you probably heard of, and I'm pretty sure you'll mention these too, is that our latest hot topics probably popping in the news and should be something we are concerned about. So let's give you the first one you probably heard about.
Avian influenza. Yeah. So flus, all these flus are kind of interesting because they're hard to get rid of, right? They a lot of times evolve. They spawn off from different animals, different sources.
Right.
And we try to incorporate, we were talking about vaccines earlier, we try to incorporate a lot of these into our like yearly flu shots, but it's not always easy because of the diversity, how often they change, they mutate, things like that, right?
Correct. And this is where some of these get a little bit dangerous, is because, yeah, you have your typical cold season, like it is probably occurring at the moment during winter season. Mm-hmm. The issue there is that there's so many strains and so many variations. You use the key term there, evolution, is that they'll switch pieces, parts of their proteins, which makes them really difficult to detect again. So even though you do get a vaccine for the latest and greatest occurring right now, next year you'll probably have to do it again, simply because they're also changing. Here the issue is that avian flu, what most people know as the bird flu, actually started causing death. It was a little bit more severe than your classic cold and flu. And so the issue is that we knew that it was being transmitted from ducks, turkeys, chickens, so your more or less poultry bit. And so people that worked in these industries started having some bigger risks. And so we then figure out that it could be transmitted from human to human, and that's where the danger began. Now, the good thing about this is that once it does jump from one human to another, it seems to kind of die off. But again, they're constantly evolving, so it's something to worry about quite a bit. Another one you probably heard of in this case?
MRSA, yeah. So in the hospital, we see that all the time, methicillin-resistant Staph aureus, right? So that's one of these microorganisms that has developed a resistance to what we would commonly treat it with. Like
correct. And so, unfortunately, because of our history of peddling drugs very often for people that just requested it, these organisms have been hit by so many of these antimicrobials and antibiotics that some of them manage to survive, and those are the ones that are resistant to those drugs. So MRSA happens to be one of the biggest contenders, simply because some of our strongest drugs, like methicillin, now happen to be kind of useless as a way of treatment. So people that are infected by these are at a much bigger risk of developing complications from these infections. Probably a fun one to know, even though hopefully most people haven't encountered it, is West Nile encephalitis, or in this case, a wonderful little virus that is transmitted typically from mosquitoes. Mm-hmm. And the concept here, it is a pretty severe version of a cold, if you will, and it pretty much can cause fatalities. So now, unfortunately here is that we haven't really found a treatment for it or even a vaccination, so we're still working on them, but it has spread out through the entire United States. We know this now, that it's happened through the transmission of mosquitoes biting birds that carry the disease, and then those mosquitoes will eventually bite you and pass on that blood bite and hit you with that disease. So this is one of those emerging diseases that is becoming more prominent. So this one here, you probably have heard about. The word bovine should be a key term. What does that tell you?
So this is the mad cow disease, right?
Right. So here, bovine spongiform encephalopathy, the key terms here is that it affects your brain. This one is going to be a very unique description. We'll spend a little bit of time talking about it, in which here we don't have a microorganism. We don't have a virus. We don't have a bacterium. As a matter of fact, we don't even have something that we consider living at this point in time. These are caused by small little proteins that are infectious called prions. And so we will discover that these guys, for some reason, can self-replicate and cause several types of brain disorders. One of the infamous ones is called Creutzfeldt-Jakob disease, and several different other cattle and farm animals that have been transmitted to humans. mad cow disease probably being the most infamous of them all. So we'll spend a little bit of time talking about that guy later on too. Now, one of the infamous ones is if you've ever had a bad day at a fast food restaurant, E. coli, but there are certain strains of them, in this case O157H7, which is a specific strain of E. coli that happens to produce toxins that in this case affect your intestinal, your colon more than anything else. And so unfortunately consuming this guy has you spend some bad days at the bathroom, more or less. So most of the ones we have are actually harmless, but there are certain variations of these guys that can cause some severe diseases. And this one still pretty much pops up 2 to 3 times per year here in the United States. Now, pretty much everybody's at least heard of this because of some movie, some show, or something like that. Mm-hmm. These are the so-called hemorrhagic fevers. These are the ones that pretty much cause you to bleed out internally. And so the Ebola virus happens to be a virus that is very, very destructive. Thankfully, it hasn't really kind of taken hold in the United States, but there's plenty of countries out there that have, in particular the continent of Africa. But the idea behind this is that it spreads rather easily and through most fluids. So fortunately, again, because of how we've managed our borders and how we've managed to actually treat the patients and help isolate the disease, it hasn't really spread too much. But this guy is fairly deadly, very quick, usually ends up killing people within 4 to 6 days. So once it starts, it's kind of difficult to stop. So this is something that is an important topic to remember. Now this one probably a little bit more hidden behind it, and the name kind of gives you the hint behind it. It's something called crypto. In this case, cryptosporidiosis comes from a protist, one of those small versions of animals that we were talking about before. This is not a bacterium. This is not a virus. This is a unicellular motile organism that, again, very similar to E. coli, causes a lot of intestinal distress, but this guy kind of likes to hide inside the folds of your intestines. And this one also pops up quite often in the United States, and it accounts roughly for about 1/3 of all the intestinal diseases that we have.
So, acquired immunodeficiency syndrome, everybody's heard of that, AIDS. So that's another virus, right? And it originally was, HIV. So it was originally transmitted from animals, right? And became human immunodeficiency virus.
Right. The hypothesis currently is that it originated from certain types of apes and monkeys more than anything else, a simian immunodeficiency virus known as SIV. Mm-hmm. And the belief currently is that somehow that virus managed to be transmitted to humans, and it stayed in humans, evolved, and now it actually has become in and of itself its own virus and its own species, right? And so since the 80s, everybody's heard the concept of AIDS, although you may not be familiar with how does it work. But HIV, the virus itself, hides inside your immune cells. And what it does is it weakens your immune system, and HIV itself is actually not deadly. But because it does destroy your immune system, that means that other organisms can kind of have a free-for-all and take you down. Mm-hmm. So because it lowers your defenses, then other opportunities occur, and that's what usually kills you. So this idea of AIDS, or acquired immunodeficiency syndrome, is not the HIV that is the bad one. It's all the other diseases that now become available to take you down simply because your immune system has been severely weakened.
And we've been working on, you said about 100 years ago they found penicillins and all these antimicrobials, and these viruses are a little bit harder to get rid of, but I know they've come out with antiretrovirals and certain things that can help at least. I don't know if they completely have found a cure yet. I know there's all kinds of testing that they're constantly doing.
Right. So thankfully we do have certain drugs that are referred to as antivirals and antiretrovirals. The idea is that these ones target the replication of the virus. And so thankfully there are certain drugs that can reduce the effect of HIV. And to this point, there are several successful cases out there in which people's viral counts, or titers is the term we use, is that's nearly to undetectable levels. Can't really quite say that, that's proof that it's 100% cured, but people can live, fully formed lives without any issue. as long as they're taking these drugs. So there is definitely lots of hope for these type of drugs. Now, one of the fun ones that is popping up very recently is for anybody that travels, especially in places that are infested with mosquitoes. So tropical areas happen to be a lot of them. Is a specific disease called Zika. And this is transmitted by the Zika virus, also by bites of mosquitoes. The biggest issue here is that, it produces a pretty bad kind of flu-like kind of cold and lots of pain. But the issue is that if it gets transmitted to a pregnant female here, is that the child typically can have some severe effects, a subject known as microcephaly, in which the skull is reduced and the actual size of the brain is also reduced. So there's a severe risk for females that are pregnant at this point in time. Its cousin, called the chikungunya virus, same idea, no issues with microcephaly, but this one has this effect that it seems to transmit just like a cold, just like Zika does too, and most other flu viruses. The only difference is that when this mosquito bites you and transmits the chikungunya virus, it seems to collect itself in the joints, causing a much more extended amount of that kind of malaise that people get, that pain, of that uncomfort or discomfort that people receive. And this one can last up to a couple of years after the infection, despite the fact that the virus has gone away. Right. So this one leaves you a mark that keeps you around for quite some time. Another aspect behind viruses is, again, an agricultural aspect, is the fact that there's viruses out there that are jumping from animals to humans. In this case, one of the common ones here is the human version of the equine virus here that severely destroys, in this case, horses and associated animals. But this is part of the farming and agricultural areas, and now it's being transmitted to humans. And so the same concept that occurs to animals is now occurring because this virus is jumping onto humans that are in constant contact with these animals. So the issue here is that it pretty much kills the animal, and now we know that it can probably do the same thing in humans. So again, risks of people that work with these is a big deal. And then probably the one that we're going to finish off with right now is probably one of the reasons why people are getting sick everywhere. And this is your classic flu virus. Now, this is a much bigger subject to deal with, but there's two groups of viruses that are usually responsible for most flu-like kind of symptoms that everybody experiences. These are the coronaviruses and the noroviruses. And one of the biggest deals that is occurring right now is the fact that there's one specific type of coronavirus called NCOV 2019 that is occurring currently in Asia, specifically in areas of China, that is transmitted pretty fast. Same virus that is associated with diseases like SARS and MERS. But in this case, the issue is that it's now causing deaths. So out of the so far several hundred cases that occurred in the last few days, now people are starting to die, and there's the small possibility that this is spreading out to several different Asian countries, and because of international travel, this is being transmitted in the United States and throughout the American continent. So there's several different reports of small cases in the United States, Mexico, and even Canada right now. So something to look out for later. So this is the field of emerging infectious diseases, or EIDs. Lots of good topics to talk about. This is why we learn microbiology. This is why this subject is important.
All right. So that is pretty much everything that we wanted to cover in this this first lesson, right? This first PowerPoint.
Right. So we've covered all 6 SLOs. Anything from the importance to a little bit of the history, as well as some of the latest and greatest diseases that are occurring. So this should kind of give you a brief introduction to what's the big deal of microbiology, not just the bad, but also the good. So we'll close off with that.
All right.